Hello everybody! Today I'm going to introduce the Pre-humanized VHH llama library developed at Antibody Studio Inc. For this robust phage VHH library, the framework of each VHH sequence was pre-humanized. The application of this pre-humanized library for novel antibody discovery can save 6 to 8 months of development work. The typical VHH discovery process starts from 2 to 3 months of immunization, 1 month of library construction, 1 month of panning, 1 month of lead selection, 2 months of lead characterization, and 3 to 4 months of lead humanization. Antibody Studio's pre-humanized library saves the time for the key time-intensive steps such as immunization, library construction, and lead humanization. This library was developed from the PBMCs of 65 llamas purchased from a llama farm, and we initially constructed a naive VHH phage library. We then performed overlapping PCRs and replaced the llama framework with humanized framework. Meanwhile, we also performed CDR shoveling. As a result, this yielded a pre-humanized library with new combinations of CDR domains, creating more diversity. New antibody discovery applying this pre-humanized VHH library can be achieved with various panning methods, either by protein panning, cell panning, biotinylated antigen protein panning, or even a combination of these methods. This is a case study highlighting the process of antibody discovery for neutralizing and blocking antibody against SARS-CoV-2. This was in response to the COVID-19 pandemic, and within a couple of months, we were able to develop the first tri-specific VHH-FC lead candidate. Here is a brief outline of the development. Step 1 shows the same library construction that I previously described. Step 2 involves the panning and screening with the recombinant S protein antigen. Step 3 describes the screening of the S binding candidates with S-ACE2 blocking function. Step 4 summarizes the combination studies of lead candidates with synergistic VHH pairing. Step 5 demonstrates the assembly of bi- or trispecific VHH-FCs based on the results from Step 4 with computer-aided design. And lastly, Step 6 shows the overall built-in mechanism of action from this novel COVID-19 trispecific antibody. Taken together, this case study demonstrates the robustness and speed of our pre-humanized VHH library. Based on the functional data, a trispecific VHH-FC was selected as the final lead antibody against SARS-CoV-2. This antibody neutralizes and blocks the viral S protein and human ACE2 receptor interactions observed both in vitro and in animal. Since this antibody binds multiple epitopes on the viral S protein, this unique design feature may better antagonize viral mutations as we have observed during the pandemic. This serves as a case study that our pre-humanized VHH library can be applied for the development of therapeutics. In addition to the trispecific antibody with neutralization and blocking of the S-ACE2 interaction, we also partnered with NYU to develop bispecific antibodies. This figure shows the following results from this collaboration. First, this heat map depicts the normalized blocking activity of VHH clones against S protein and ACE2, as well as other potentially new receptors on myeloid cells. Figure C shows the kinetics of the monoclonal and bispecific VHH-FCs with good affinity. Figures D, E, F, and G demonstrates various functional assay data. Overall, antibodies developed from AB Studio VHH libraries have exhibited wide-ranging diversity. We have demonstrated our studies in these following publications, and if you are interested, please feel free to request the PDF files from us. This is another case study in which we applied the pre-humanized VHH library and performed cell panning against two separate cancer targets, CD38 and BCMA, to develop a novel trispecific antibody to serve as a T-cell engager. Again, this demonstrates that this robust VHH library can be applied for a wide range of therapeutics, including cancer. Our case studies are shown in this table. Thus far, we have successfully completed about 15 projects using our pre-humanized VHH phage library for antibody discovery and development. We look forward to opportunities to support more companies to develop humanized VHH antibody therapeutic candidates. Thank you very much. If you have any questions, concerns, or inquiries, please feel free to email us at abstudio.info@antibodystudio.com.